Okay. Good afternoon, children. The topic which is given for me to discuss is principles of inheritance and variation, which is considered to be one of the important topic for NEET, and maximum around say 2 to 5 questions from this chapter will be appearing. So it is considered to be one of the most important chapter. So today we will discuss on the synopsis for this particular chapter. Later we will discuss on the questions related to this particular chapter. Okay. So the chapter that we are going to discuss today is principle of inheritance and variation. Okay. What we are going to do here today is we are going to discuss some of the important terms which is related to this particular topic. Okay. The first term you have to understand here is you should know what is genetics. Second, you should know what is heredity. Next, you should know what is inheritance. Okay. First, we will discuss the meaning of these three particular terms. Okay. So, what is genetics? The word itself very clearly say genetics is the study of heredity, or I can use the word inheritance. So it says genetics is the study of heredity and inheritance. Now, what is heredity? Everybody would have studied. Heredity is nothing but transfer of character from, I can use the word parent to offspring. Instead of offspring, I can also use the word young ones. Or if you want me to simplify further, I can use the word children. Okay. So, when I use the word heredity, you should understand I am talking about transfer of character from parent to offspring. The other name of offspring is young one, or I can use the word children. Now, what is inheritance? Inheritance is the process of, please understand, inheritance is the process of heredity. Okay. What is inheritance? Inheritance is the process of heredity. That means how a particular transfer, how a particular character transfer from one generation to other generation. Understood? No? Again, I'm telling you, inheritance talks about how particular character transfer from one generation to other generation. The process is called as what? Inheritance, and the actual is called as heredity. Hope you understand, children. So, what is heredity? Heredity is nothing but transfer of a particular character from parent to the offspring. It's called as heredity. And what is the process in which the heredity happens? Which is nothing but the inheritance. I hope you understand. Can we go to the next important terms? Here. Okay. We'll talk about the next important terms. Okay. The first one, you should know genes, chromatin, chromosome. Okay. And you should also know about character. Right. Okay. We'll see the definition for each of these words, and then some more terms are there. We will see that later. Okay. First one, what is a gene? What is a gene? Textbook says gene is a unit of inheritance. Okay. What is a gene? A gene is a unit of inheritance. Okay. Is this enough to understand a gene? No. Much more you should know about the gene. We'll see one by one. Okay. Now, hope you remember DNA. Okay. Hope you remember about DNA. You would have studied in your first PUC in biomolecules. You would have studied about the structure of DNA. Okay. So, what is a DNA? We studied DNA is a double helix thread-like structure. Okay. Imagine this is a DNA. What is this? A DNA. Now, you know very well, in DNA, there are certain segments, not all. There are certain segments in the DNA. Understood? There are certain segments in the DNA. Now, these segments are very special. You can ask me why these segments are very special. Because these segments have a specific information. What these sections are having? These sections are having a specific information. Now, the specific information for what? The specific information for protein synthesis. Okay, they have information for what? Protein synthesis. And this protein is the one will express or appear as a character. Understood? Again, I'm repeating. Imagine this is the entire DNA. These DNA have certain segments. Okay, not all. Only the segments of DNA has a specific information. Now you can ask what information? Information about what? Protein synthesis. And this protein which is synthesized will be expressed as a character. Okay. So whatever character a person is having, okay, maximum, whether he is tall or short, whether he is having blue color eyes or red or black color eyes, whether he is having long hair or short hair, all these characters are due to what? Protein, an expression of a protein. And why, from where this protein is synthesized, the information come from? The information come from a specific segment, and this specific segment of DNA is called as genes. Understood, children? So, what is the definition of a gene? So, I can say a definition of a gene, I can call it a segment of DNA. So, can I write like this? A gene is a segment of DNA. Yes or no? A gene is what? A segment of DNA. Why this segment is so important? This segment is so important because it has a specific information. Specific information for what? For protein synthesis. Now, why protein has to be synthesized? Because this protein will be expressed as a character. So, what is a gene? A gene is a segment of DNA which has an information for protein synthesis, and this protein will be expressed as a character. So there is two definitions for a gene. Number one, gene is a unit of inheritance. Second, a gene is a segment of DNA which has information for protein synthesis. The protein will be expressed as character. Understood now, children? So what is a gene? If anybody asks what is a gene, you can say it is a unit of inheritance, or you can say it is a segment of DNA which has specific information for protein synthesis, and the protein will be expressed as what? Character. So hope you understood about genes. Okay. Let's talk about chromatin. Let's talk about what? Chromatin. Okay. I'll take this color. What is it? See, for example, I told you this is DNA. Imagine this is DNA. Now, this DNA, I am coiling it. This DNA, what I'm doing? I'm coiling it with the help of certain proteins. Okay, so what are these? These are protein. What are these? These are coiled DNA. Okay, so this is DNA. Now, if I, with the help of protein, when I coil the DNA, now this become chromatin. Understood? Now, if I further condense it, if I further condenses, this becomes chromosome. Okay. So the next one is what the chromosome consists of. The chromosome consists of chromatin. And what the chromatin consists of? Chromatin consists of DNA. Now, what this DNA consists of? The DNA consists of genes. Understood? Clear? So, DNA consists of gene. When I coil the DNA, it becomes chromatin. When I further coil the DNA, when I condense the DNA, it becomes chromosome. So, chromosome is actually made out of what? DNA. Now, the next question, where the gene is present? I can say gene is present in the DNA. Gene is present in the chromatin. Gene is also present in the chromosome. Understood? So, we studied about DNA, chromatin, chromosome, and what is a gene. Okay, again, I'm telling you what is a gene. A gene is a segment of DNA which has information for protein synthesis. Now, where the genes are present? The genes are present in the DNA. When the DNA becomes little coiled, it becomes chromatin. When the DNA becomes further coiled, it becomes chromosome. Hope you remember the structure of chromosome. The chromosome consists of centromere, and the chromosome consists of chromatid. Okay, how many chromatids are there? There is one chromatid is present and one centromere is present. Understood? No? So one chromatid and one centromere. Any doubt? Okay. Now, listen carefully. Now, imagine this is a chromosome. How many chromosome? One chromosome. Now tell me, we will use different color for the centromere. Okay. Now tell me, this is one chromosome. This is how many chromosome? This is not two chromosome. This is also one chromosome. This is how many chromosome? This is also one chromosome. How I know all these are one chromosome? Because if a centromere is one, understood? If centromere is one, the number of chromosome is also one. Let it be one, two chromatid or three chromatid or four chromatid. Okay, the chromatid number may different, but if the centromere is one, it is considered to be one chromosome only. Understood? So here I can say this is one chromosome consists of one chromatid. Here I can say this is one chromosome consists of two chromatid. Here I can say one chromosome consists of four chromatids. So number of chromatids may different, but if the centromere is only one, it is considered to be one chromosome only. That you have to understand. Okay. So I can say the number of chromosome, if you want to count, if the question comes, if they give the picture and ask you to identify how many chromosomes are there, all you have to see is what? Chromatid. If number of centromere, I'm sorry, if number of centromere is equal to number of chromosome, remember this. Okay, you have to understand this. Okay, so we discussed what is a gene and where the gene is present in the DNA, what is a chromatin and what is a chromosome, and how to find out the number of chromosome. How you will find out the number of chromosome? The number of chromosome is equal to number of centromere. Clear? Can you go to the next one? So what and all we discussed? We discussed about gene, we discussed about chromatin fibers, we discussed about chromosomes and how to find out the number of chromosomes. Okay. Okay. Related to this particular topic, let's talk about cell cycle. Hope you remember the cell cycle consists of G1, S, G2 and M phase. Okay, there is one more phase is called G0, which we are not going to talk about. We are going to talk about G1, S, G2. Okay. Hope you remember during S phase, the DNA will be doubled. Otherwise, I can use the word DNA will be replicated. That means one DNA, imagine if one DNA is there, it becomes how many DNA? 2 DNA. One DNA becomes how many DNA? It becomes 2 DNA. Okay. So, what you understood here, you should understand the DNA will become doubled. But you should understand chromosome number remains the same. Please understand chromosome number remains the same. Okay. Imagine this is a chromosome and the chromosome consists of a DNA. Okay. Imagine, okay, during S phase, what will happen? The chromosome will be get doubled. Sorry, I'm sorry. The DNA will be get doubled. Okay. But the number of chromosome will be the same. See, look at this. How many DNA is there? One DNA, two DNA. So this is one DNA and this is two DNA. But how many chromosomes are there? There is only one chromosome. So during S phase, please understand children, DNA, the number of DNA will become doubled, but the chromosome number remains the same. How I know this is only one chromosome? I know this is only one chromosome because it consists of only one centromere. Understood this concept? Okay. Again, I'm telling you, in a cell cycle, during S phase, DNA replication takes place. When DNA is getting replicated, one DNA become two DNA, but one chromosome remains one chromosome only. Okay. Chromosome number will not be get doubled. Only the DNA will be get doubled. So keep that in your mind. So we spoke about gene, we spoke about chromatin, and we also discussed about chromosome. And we also found out how to identify the number of chromosome and what happened in the cell cycle during S phase. During S phase, the DNA will double, but not the chromosome number. So the next important term that you have to understand is allele. Okay. What is an allele? Definition says allele is the different form of a same gene. Okay. For example, if you take gene I, which is responsible for the blood group. Now, this gene I has three different forms. For example, IA, IB, and small I. Okay. Now, what are these IA, IB, and small I? IA, IB of small I are the different form of a same gene called as what? Gene I. And all the same gene are present in the same locus of homologous chromosome. Okay. Listen carefully. What is an allele? An allele is a different form of a same gene. Where they are present? All they are present in the same locus. And where it is present in the homologous chromosome. Okay. So first you should understand what is an allele. Just remember, allele is a different form of a same gene. Let's talk about what is a locus. What is a locus? A locus is a specific site. A locus is a specific site. See, for example, imagine this is the chromosome as a chromatid. Okay. Imagine they have certain area. Okay. Why this area is present? Very special, because this area consists of a gene. For example, gene A is present, gene B is present, C D is present, E is present, D and E is present. Understood now? So what you understood here, in a chromosome, there are certain area where a specific genes are located. Therefore, this area is called as locus. Understood? So what is a locus? A locus is a specific site where a particular gene is present. Now the next question is, what is this homologous chromosome? Hope you remember every chromosome as a pair. See, for example, imagine this is chromosome number 1. Okay. And this chromosome number 1 will have a pair. Okay. So imagine this is chromosome number 1. This is also chromosome number 1. Why? Because this chromosomes come from father and this chromosomes come from a mother. Okay. This is called as maternal chromosome. This is called as maternal chromosome. That is one from father and one from mother. So these two chromosomes are called as what? Homologous chromosomes. Understood? I hope you understand. An allele. An allele is a different form of a same gene present in the same locus of a homologous chromosome. So in this particular thing, we learned 3 words. Number 1, what is an allele? We studied allele is a different form of same gene. Where it is present? It is present in a specific locus. What is a locus? A locus is a specific site in a chromosome where a particular gene is present. And then we studied what is a homologous chromosome. A homologous chromosome is a pair of chromosome, same chromosome. One comes from mother and the other one comes from father. Okay. So 3 words we studied. Number 1, allele. Number 2, locus. Number 3, homologous chromosome. Any idea? Any doubt? Can you go to the next one? Sure. So I hope there is no doubt so far what we discussed. Okay. Again, we'll recollect. We'll discuss about gene. We discussed about chromatin fiber. We discussed about chromosome. We discussed about allele. We discussed about locus. And we also discussed about homologous. So, clear? Okay. Every gene has 2 alleles. Okay. One may be a dominant allele. The other one may be a recessive allele. We'll see this, what is dominant allele and recessive allele. Before that, we'll discuss what is the character, because these are the terms we are going to use in this particular chapter. and what are traits. Okay. Hope you understand. Character is a common feature. Character is a common feature. Trait is unique variation. Let's discuss what is this. Okay. Character is a common feature. Trait is a unique variation. What is the difference? Simple. See, for example, when I use the word height of a plant, so the word height is a character. Understood? But when I specifically say this particular plant is tall, or if I specifically say this particular plant is short, the other name of short is dwarf. Understood? So if I say a particular plant is tall or a particular plant is short or dwarf, I am actually talking about the trait of the plant. Understood the difference? So what is a character? A character is a common feature which talks about the common character. But what is a trait? A trait is a unique variation. That means it specifically talk about whether a particular plant is tall or short. See, for example, I am using the word eye color. Now tell me, eye color is a trait or a character? Obviously, it is a character because generally it talks about. So eye color is a character. But if I say that particular person is having blue eyes, or the particular person is having brown eye, then I am talking about trait of an individual. You understand? So what is a character? Technically use the word, a character is a common feature. Now what is a trait? A trait is a unique variation. Understood? See, for example, I am telling eye color, height of a person, weight of a fruit, red flower. Now, out of this, which is a trait? Which is a trait? Obviously, which is a trait? Red flower is the trait. Okay, because it specifically talk about the color of the flower. So this becomes what? Trait. Now, what are these? Eye color, height, weight. They are generally talks about. So they are considered to be what? Character. Hope you understand. Okay. So a character is a common feature. A trait is a unique variation. Clear? Can you go to the next one? Okay. Before that, let's rub this. Before that, I told you every gene will have two allele, but there are certain exceptions. We'll see that later. Every gene will have two allele. Most of the time, it may be a dominant allele, or we can use the word recessive allele. Understood now? So there are two alleles are there. One is called dominant allele. The other one is called as recessive allele. How it can be represented? Dominant allele can be represented by the capital letter, and recessive allele can be represented in the smaller letter, small letter. I hope you understand what I'm talking. See, for example, tall, I want to say capital T, and recessive, see, for example, tall is a dominant character, so I represented in capital T. Short is a recessive character, I represented in small t. Understood? But I should not write only one t. I am supposed to write capital T, small t, or I can say capital T, small t. Now, question is why I am writing two times? I am writing two times because you should understand for every character, there are two alleles are involved. One should come from father, the other one should come from mother. Understood? Therefore, for every character, I should write how many times? Twice I should write. Clear? So, dominant character, I should use capital letter. And how many times I should use? I should use twice. Recessive, small letter, how many times I should use? I should use twice. Now, what is a dominant allele? A dominant allele will always express. Okay. What is a recessive allele? Recessive allele will be expressed, listen carefully. Recessive allele will be expressed, listen, only in the absence of dominant allele. Okay. So, what is a dominant allele? A dominant allele will be always be expressed. A recessive allele will be expressed only in the very important absence of dominant allele. Clear? Next, dominant allele will be expressed in both the condition. Now look at this. I can call this as capital T as a homozygous condition. Why homozygous condition? Look at this. Two alleles are same. Otherwise, I can use the word capital T small t. I can call this as heterozygous condition. Now, what is heterozygous condition? Now, look at this. Hetero means different. Homo means same. Hetero means what? Different. Now, what is different here? Hope you understand. Two alleles are different. Therefore, I will call this condition as a heterozygous condition. So, a dominant allele will be expressed both in the homozygous condition and in the heterozygous condition. What is a homozygous condition? Both the alleles are same. Heterozygous condition, both the alleles are different. But when you look at the recessive, recessive will be expressed, look at here, small t, small t. Now tell me what is this? Is homozygous or heterozygous? Yes, it is called as homozygous condition. So, recessive allele will be expressed only in the homozygous condition. Only in homozygous condition. But dominant will be expressed both in what? Homozygous condition and heterozygous condition. So, in this, we also learned two more term, what is homozygous and what is heterozygous. Understood now? So, a gene will have most of the time, not all the time, most of the time will have two alleles. One is called a dominant, the other one is called as recessive. Dominant, you should represent in the capital letter. Recessive, you should represent in small letter. Dominant will be expressed both in homozygous condition and heterozygous condition. Recessive will be expressed only in the homozygous condition. Dominant will always be expressed, but recessive will be expressed only in the absence of dominant allele. Hope you understood this concept. Let's go to the next concept. Okay. Let's talk about the haploids and diploids. Okay. Let's have one more term which is related to this concept: haploid condition, diploid condition. Okay. Haploid condition, listen carefully. Haploid condition. Haploid condition is half the set of chromosome. And diploid is the complete set of chromosome. It's a complete set of chromosomes. So, for example, in human, when I say 23 chromosome, then it is become haploid. But when I say 46 chromosome, it becomes diploid. Understood? So it's this everybody knows. Okay. But when you're writing a chromosome, say for example, I'm sorry, when you're writing an allele, listen carefully. Say for example, I'm writing capital T, capital T, capital R, capital. Okay. Now tell me what is this condition is. Okay. Now capital T, capital. Okay. So two condition I have written here. Okay. Here two are there. So is this considered to be diploid? No. Two are there, but listen carefully. But both are see different. See for example, I am writing capital T, capital T, capital R, capital. Now tell me in this which are diploid and which are haploid. Now can you tell me this is diploid or haploid? Yes, it is diploid. You can see capital T, capital T. Same are there. So this is diploid condition. Now tell me what is this condition? Capital R, capital R. What condition? Diploid condition. Now again I am writing capital R, small r, capital T, small t. What condition they are? Hope you understand. This is also what condition? Diploid condition. Why? Because alleles are same. Now what is this condition? Two same allele, two same allele. So this is considered to be what? Diploid. Now what is this? Two alleles are there. Can I consider this as diploid? No. Why? Because these two alleles are different. So this condition should be called as what? Haploid condition. Why this is haploid condition? Because it has two different allele and how many times represented? Only one time represented. So it is called as what? Haploid. Okay. Now tell me capital T, capital R, capital Y, capital T, capital T, capital R, capital R, capital Y, capital Y. Okay. Capital R, capital R, capital Y, capital Y, capital R. Okay. Now remember. Now the question comes. Identify whether it's diploid or haploid. Hope you remember. Can you tell me this is diploid or haploid? Correct. This is haploid. Why? Because you can see three different alleles are there and all three different alleles are represented only once. So this is considered to be what? Haploid. Now look at this. Three different alleles are there, but three different represented twice. So this is considered to be what? Diploid. Now this is diploid. This is haploid. This is haploid. Understood? Okay. This can also be called triploid. We'll see that later. But now you understood this. How many times is represented? Based on that, it is considered to be whether it is diploid or whether it is haploid condition. I hope you remember what is this diploid and haploid conditions are. Okay, the last. Okay, let's talk about the multiple alleles. Alleles we studied. I'm sorry. So like another 5-10 minutes, I'll finish. Multiple alleles. Okay, we studied alleles. Now multiple. We studied every gene generally will have two alleles. Am I right? And these two alleles may be a dominant allele or we studied it is a recessive allele. We also discussed what is dominant allele and what is recessive allele. Okay. Now, one more concept is called as multiple allele. Simple. For example, if a particular gene will have more than two alleles, then that particular condition is called as multiple allele condition. Example I already given, blood group. Okay. The gene which is responsible for the blood group is gene I. Now, this gene I has three different alleles we studied. Okay. One is called IA, The other one is called as IB. The other one is called as small I. Why small I? Because this is recessive. And why capital A, capital B? Because both are dominant. Now, when you look at this gene I, now the gene I is having how many alleles? 1, 2, 3. How many alleles are there? 3 alleles. That means more than 2 alleles are there. So this condition is called as what? Multiple alleles, and the concept is called as multiple allelism. So if the question comes, give me an example for multiple alleles, then you can write blood group. To be precise, you should write ABO blood group in humans. Okay, so this is multiple alleles. Any doubt? So we discussed about multiple alleles. Okay. The last concept, we study what is genotype and phenotype. With this, we'll finish the introduction part. And in the next class, we will discuss on the questions based on this. Okay. First one is genotype. The second one is phenotype. What is a genotype? Genotype. Genotype is a genetic makeup. What is genetic makeup? The genotype will tell me whether a particular allele is present in the homozygous condition or it is present in the heterozygous condition. See, for example, if I write capital T, capital T, capital T, small t, this capital T, capital T, capital T, small t is nothing but a genetic makeup, and the genetic makeup is nothing but a genotype. What is a phenotype? Phenotype is the external appearance. See, for example, when I say capital T, it's for tall character. Capital T, small t is also for tall character. And small t is for, I can say, a dwarf trait. Now, this tall and dwarf, okay, this external appearance is called as what? Phenotype. Okay, so genotype and phenotype. Genotype is a genetic makeup, and phenotype is the external appearance. Otherwise, I can use the word morphology of a particular species. Okay, the last one. Okay, the last concept. What is a test cross? Okay, a test cross is F1 hybrid along with a recessive trait. Now, capital T, small t is called as what? F1 hybrid. Hope you remember the monohybrid cross. When they crossed capital T crossed with small t, small t. In F1, what you will get? You will get capital T, small t. Okay. Now, this capital T, small t is crossed with small t, small t. This is considered to be one of the test cross. Okay. Now, when you do this cross, hope you remember the gamete formation. Okay. So, this will give you capital T, small t. This will give you capital T, small t. This will give you small t, small t. This will give you small t, small t. So what is the ratio? Ratio is tall is to dwarf. I can write 2 is to 2 will be reduced to 1 is to 1. So a test cross is a cross between F1 hybrid. Otherwise, I can use the word unknown individual. Okay, that means you don't know the genotype of it. Only the phenotype you know. Okay, unknown individual, or I can say it is a cross between F1 hybrid with a recessive parent, or it is a cross between an unknown individual with a recessive individual. And what should be the ratio? 1 is to 1. So the test cross ratio is always what? 1:1. Remember this. Okay. So we discussed what is a test cross. Again, I'm telling you, test cross is a cross between F1 hybrid and the recessive parent, or cross between an unknown individual with recessive individual. Understood? That is called as what? Test cross. And the ratio of the test cross is 1:1. Okay. So with this, we'll stop. Okay. So these are the important concept that you should remember in principles of inheritance and variation. In the next class, we will be solving the questions. When we solve the questions, you understand the concepts better. Thank you.